Now continuing from here, we are moving to the second stage, which is the metaphase stage. Metaphase stage. This much we have learned that in metaphase stage, number one, I am writing the key events. Alright. Metaphase, condensation of chromosomes. Is complete. So what does it mean? They are the thickest and shortest in this phase. Right? Second point, the spindle formation. Remember that we discussed that it starts at one pole and it moves to the other pole. The spindle formation is complete. And since it is complete, all the chromosomes lie at the equator. Okay, where do the chromosomes lie? They lie at the equator. What does it mean? It means one chromatid of each chromosome is attached to the kinetochore. I hope you remember kinetochore. This is the link. Remember, I showed you in my previous video that these are disc shaped structures which will bind to the spindle fiber. So one chromatid of each chromosome, one chromatid of each chromosome, it will be attached by the kinetochore to the microtubules from one pole. By the kinetochore to the microtubules from one pole, while its sister chromatid is attached by its kinetochore to the microtubules of the opposite pole. Alright, so what are the key features? Spindle fibers attached to the kinetochore of the chromosome and chromosomes are moved to the spindle equator, gets aligned along the metaphase split through the spindle fibers to both the poles. How is this arrangement seen in a diagram? When we draw the diagram, we say that this arrangement is mainly seen as this is the cell where the spindle apparatus, that means the centrioles are present, astral rays, that is what I am drawing here. Same way on the other side, astral rays. Clear? These are the spindle fibers. To which these chromosomes, these lie at the equator, forming an equatorial plate. So once these lie at the equator, they form equatorial plate, or we call it metaphasic plate. This is the identifying feature of a metaphase stage. So what do they form? Metaphasic plate or equatorial plate. Clear? This is what is the key feature, that chromosomes are moved to the spindle equator get aligned along the metaphase plate through the spindle fibers to both the poles. Clear? Moving to the third stage, the anaphase stage. In this anaphase stage, now the spindle fiber breaks. This one spindle fiber. Okay. First one is spindle fiber breaks. Break and the centromere split. Centromere, to which the kinetochore discs are attached and chromosomes are on the above, that is at the top and the bottom. So this centromere, it will split. Clear? What will happen if this will happen? This is going to split. Half chromosomes along with half centromere along with one chromatid is now called the daughter chromosome. Daughter chromosome. We cannot say daughter cell. Still it is happening in the nucleus itself. We cannot say it is cell. It is just a daughter chromosome. Now it will start migrating towards the pole. This daughter chromosome from each pair begin the migration towards the poles. So key events are centromere will split and chromatids will begin their migration towards the poles. How will it appear in the diagrammatic view is, let's change this particular one into the diagrammatic version. We are now no more talking about metaphase, we are talking about anaphase. So automatically this has to go, right? Whenever these chromosomes are moving, first of all, the length of these spindle fibers is not at all fixed. And the shape of the chromosomes, it can be inverted I, L, V and J. Okay? I, V, L and J. It could be in any shape. So, basically we have to look for this thing, that whenever the centromere splits and the chromatids separate, they commence their movement towards the opposite poles. They show various shapes. Shapes are I, V, L and J. So these are the shapes in which all the chromosomes commence their movement towards the opposite pole. Clear? Now moving to telophase. After this, the entire material will reach to the opposite poles and will get further decondensed. Condensed is, it is becoming thick and chromatid chromosomes. Decondensed is when it is going to become fibers once again. Now the last phase, telophase. In this telophase, first of all, chromosomes have reached their poles, and what will they do? Chromosomes start decondensation. Alright, become thin and long. They lose their individuality. This is what it means. Clear? Secondly, it is done. Thirdly, nuclear membrane. Remember which disappeared? Nuclear membrane, nucleolus, Golgi complex, and endoplasmic reticulum. What will they all do? Reappear. Okay. Now, basically what is happening? Chromosome clusters, they will reach the poles and identity is lost. Nuclear envelope, it assembles around the chromosome and automatically nucleoli, Golgi, endo, these all are formed. So, how will it look like? It will somewhat be like this. That now in a cellular apparatus, these cells will show that again this is also showing disintegration a bit because the spindle fibers are no more required. So disintegration, this will also show disintegration. What is this called? Spindle apparatus. So it will show disintegration. And nuclear envelope is completely getting formed where the nuclear material further once more getting condensed to form, decondensed to form fibers, and these are thin and long fibers. Nucleoli, then endoplasmic reticulum, Golgi apparatus, this all is showing that it is again being formed. Then once this happens, what will happen? The next step is cytokinesis. That means now the division of cytoplasm has to take place. Why will it happen? Because two daughter nuclei are formed which are now being separated into two daughter cells. Okay, ultimately these two nuclei will get a cytoplasmic sheath around and will form two daughter cells. And the process differs in animals and plants. We call it cytokinesis in plants and cytokinesis in animals. Clear? That we will discuss in detail, but first of all we have to discuss the significance. What is the significance of this entire process? It means after the telophase, what we have received are two daughter cells, clear, with a little cytoplasmic integration material with proper nuclear material well separated and condensed, decondensed chromosomes, long and thin fibers. These are there. It means we get two daughter cells now from one cell without reducing its DNA content. Okay, so what is the significance of mitosis? First point, in multicellular organisms, body growth takes place by mitosis. Number 2, replacement. Remember, repair, replacement of worn out cells, worn out tissues also, like blood cells. When the skin slots off, so skin cells, these are getting replaced. Number 3, repair. Repair of what? Injured tissues. You can make it 1 point also for repair and replacement. Then number 4, in unicellular organisms, This is the mode of asexual reproduction. Means unicellular organisms, mitosis is involved in the asexual reproduction. Then number 5. What about plants? Remember we discussed in the plant tip, root tip, shoot tip, this happens in flowers, in fruit formation, it happens. So over there also, vegetative propagation, it is entirely dependent on mitotic division. And what it forms then? Genetically identical twins. But when so many things are going on in significance, we should also discuss that what is not going according to what is expected. It means if uncontrolled cell division takes place in certain tissues, or organs, it will result into tumor. Okay, this is a kind of contact inhibition which is seen in these cells, that they stop their own growth. But when there is no stoppage, it means uncontrolled cell division going on and on, we call it tumor or cancer. Clear? This is what is about M phase or mitosis.